Hello everyone, I'm Debdatta from Dr. Dong's group. Today I'll be talking about my project on development of a pH-responsive peptide-based hydrogel for effective eradication of biofilm, one of the most well-known bacteria that affects wounds and causes nosocomial infections is the methicillin-resistant Staph aureus, or MRSA. The biofilm produced by this bacteria makes it resistant to conventional antibiotics and increases its pathogenicity. The treatment of MRSA-infected wounds using hydrogels offer a novel alternative to conventional therapeutic means. Hydrogels are cross-linked polymeric networks capable of retaining large amounts of water and maintaining its 3D hierarchical structures. Therefore, hydrogels are capable of encapsulating antimicrobial peptides to treat infected wounds. Moreover, hydrogels can mimic the extracellular matrix, thereby supporting tissue regeneration and healing while eradicating biofilm-forming bacteria. In our lab, we have created a pH-responsive peptide polymer hydrogel. The hydrogel dissociates at low pH, which is often found in chronic wound sites. Upon disassembly, the encapsulated antimicrobial peptide is released, which disrupts the bacterial biofilm, aiding in the healing process. The pH-responsive peptide was designed by substituting one of the hydrophobic leucine residues in a previously reported hydrogel-forming peptide called 141 with a non-natural amino acid FMOC-XP synthesized in our lab. The pKa of a tertiary amine usually lies in the basic range. However, when it is tucked inside the core of a beta sheet structure, such as is formed by the self-assembling peptide called 141XP, the pKa then shifts to a neutral or weakly acidic range, which is biologically significant. Further, this amino acid residue renders better pH responsiveness to the peptide due to its high hydrophobicity and ionizable nature. It can be easily accommodated in the tight hydrophobic interior of the peptide self-assembly without entropic penalty, unlike natural basic amino acids like lysine or arginine. The hydrogel was prepared by mixing an aqueous solution of the peptide with a polymer called polyethylene glycol, PEG. Strong gelation resulted from the self-assembly of the peptide in addition to the covalent bond formation between the peptide amine groups and the NHase group in the polymer. Further, a phosphate ion containing buffer was added, which stabilized the gelation through electrostatic interactions. The strength of the hydrogel was assessed by the high storage modulus observed in rheological studies. Moreover, when a 1,000% shearing strain was applied, the hydrogel recovered its modulus after removal of the strain, depicting its self-healing property and ability to be used as injection. pH responsiveness of the newly designed 141XP peptide was proven in CD spectra, where we observed characteristic beta sheet peaks at pH 7.4 and random coils when the pH was lowered. This was also observed in TEM images. A similar trend was observed when we conducted these experiments on the peptide polymer hydrogel. pH-triggered disassembly of the hydrogel was further assessed in a rheology assay where the hydrogels were incubated in different pH buffers prior to the experiment. Expectantly, the storage modulus was much higher for the gel incubated at higher pH. Following this, we assessed the ability of the hydrogel to encapsulate a previously developed cationic rhodamine-labeled antimicrobial peptide through a time-dependent release assay. As you can see in this graph, the release rate was much higher at the lower pH, suggesting disassembly. Antimicrobial activity of the hydrogel was assessed using a live and dead assay on MRSA biofilm. We observed a higher number of dead bacteria stained red at pH 5.5 compared to the live bacteria, which took up the green stain. The higher bacterial killing efficiency of the hydrogel at lower pH was also observed through a bacterial plate counting assay. Moreover, we found out that the hydrogel was highly compatible with mammalian cells with an average cell viability of 97%. In conclusion, we have demonstrated the design and synthesis of a pH-responsive hydrogel capable of encapsulating antimicrobial agents and releasing them in a pH-dependent manner. This controlled release ensures targeted delivery and lower cytotoxicity, providing a safer treatment option for patients with biofilm-associated infections. In the future, we hope to expand the library of pH-responsive hydrogel-forming peptides to encapsulate drugs like insulin for the treatment of diseases such as diabetes. Finally, I would like to thank my research advisor, Dr. Dong, and my lab mates for this project. I would also like to thank UT Arlington and NSF for aiding our work. Thank you all for listening to my talk, and I look forward to answering any questions you might have.